Okay, Jack, I think you can start whenever you want.
Okay, well, let me share the screen first, and here we are. Can you see the presentation?
Yep.
All right, so welcome back to the session on quantum biology. This is the fourth and last installment, hopefully the most interesting. In the first installment in this series, we talked about the general principles of quantum physics. Then we spent some time looking at why biology might benefit from quantum phenomena and what are the challenges and sort of general dimensions of biological systems, timescales, and whether quantum processes in general apply. Then the third session, last week, we looked at some specific examples of quantum biology applications, effects ranging from photosynthesis to olfaction and quantum metabolism. And today we will look at things which are a bit more speculative, but maybe for this reason a bit more interesting. And all of this will be focused on aspects of neuroscience, including anesthesia and consciousness. And we will start with the soft introduction into what our human brain is all about and why is it so special. Try to answer some profound questions, such as what is consciousness and where is memory stored? We don't even know that for certain. And as a physicist, I'm interested in some performance indicators of our human brain, and in particular, the computational power of the brain, energy requirements, the processing time, and the number of bits we can process per unit of time. And then this will lead to the question whether this processing is actually classical, quantum, or maybe something else altogether. None of the above. And it's quite interesting. So let's start with some numbers which are pertinent to this topic, the human brain by numbers. So first of all, we know that the elementary units of processing units in the brain are neurons, and there's about 100 billion neurons in the human brain on average, which is a staggering number. But it's even more staggering when you think about how many connections each neuron makes with other neurons by synapses. And that ranges from 10,000 up to 100,000 synapses per neuron, which gives us something like at least 10 to the 15th synapses. And if we conservatively assume that they process about 10 impulses, action potentials, electrical impulses per second, then that gives us at least an order of magnitude of the number of operations, whether they are all information processing operations or something else. It's hard to say, but in principle, this conservative number is 10 to the 16th synaptic operations per second in the brain, which is a staggering number. It can be compared to the number of operations of the currently state-of-the-art supercomputer clusters, such as the Blue Gene, and which has performance about 10 to the 15th, so an order of magnitude less. And I will argue that actually 10 to the 16th operations is a lowball estimate. So that's comparable. And of course, we know that supercomputers have been better than some of the best humans in some specific tasks, such as the chess, the famous deep Blue Gene computer competing with Garry Kasparov, I believe it was, at the Grandmaster champion chess, and even more recently playing Jeopardy. But all of these are individual, specialized, single-purpose computational algorithms, which are, of course, outperforming humans or maybe performing at similar levels to the best of our competitors in these areas. However, we have to keep in mind that the human brain is performing many other functions at the same time, not just, let's say, playing chess or playing at Jeopardy. We do many more things, and the brain is coordinating all our functions, bodily functions, physiological processes, registering a lot of inputs, visual, tactile, audio, and so on. So that has to be taken into account when comparing the supercomputers with our brain. And actually, even more impressive is the fact that the brain operates at a very low level of power consumption compared to the supercomputer. The blue gene requires something like 1.5 megawatts, which is the power supply for the city the size of Red Deer or so, maybe a bit smaller, but anyway, 50,000 or so people. So it's a huge amount of power that is provided for the purpose of computational operations by the supercomputer. By comparison, the human brain only requires about 25 watts. Although this is 25% of the physiological demands of the human body, of about 97 watts on average to about 100, but 25 watts is the power required for a small bulb in the night lamp, and actually all of 25 watts is not just for computational or cognitive processes of the brain, but also everything else that goes with it, such as maintaining temperature, building proteins, providing blood supply, and all of this. So perhaps a fraction of it is utilized only for cognitive and computational processes. Let's say maybe 10% at most. I would say 3 watts, which is what you need for an electric toothbrush, basically. So when you think about this, is absolutely incredible that such a low amount of energy is sufficient to perform such complex functions as solving differential equations, crossword puzzles, or playing chess at a high level. All right, so the structure of the brain is quite complex, but I will focus on the structure of the neuron, which, as I mentioned, is the basic unit involved in computations in the brain. Some people argue that the brain is not a computer at all, but definitely we can compute. So part of the brain's activity is computational, and here is on the right is a cartoon representation of the neuron with the prominently displayed microtubules, of which we heard a little bit in the previous lecture. The thick lines are the microtubules in the so-called soma, the body of the neuron. In the middle, we have the nucleus, and then we have the long protrusion called the axon, which is actually packed with microtubules, and they are interconnected with microtubule associated proteins, which play a major role in stabilizing this whole network. And the word network here actually is very appropriate. In fact, the person who is mentioned here at the bottom left, Stuart Hameroff, wrote a kind of prophetic book in 1987, so a long time ago, 34 years ago, which was called Ultimate Computing. And in this book, he argued that actually microtubules in the neurons are the ultimate computational devices. And we are still chasing that metaphor today, trying to understand what they can and cannot do. but they can do a lot, and I'll say a few words about that. Microtubules and microtubule-associated proteins form this parallel computing-like infrastructure of the neuron, especially in the dendritic part of the arbor of the neurons, where synaptic connections play a major role in signal transmission between neighboring neurons. And as I mentioned to you a few minutes ago, we have tens of thousands of these connections per neuron. So that's an incredible amount of interactions taking place at the same time. Now, we'll also discuss later on briefly, of course, because we don't have that much time. That's maybe something for a semester's worth of lectures. Things like neurodegenerative diseases, Alzheimer's, various dementias, bipolar disorders, schizophrenia, and so on, which pretty much all of them have something to do with the impairment of the cytoskeleton and microtubule damage or secondary aspects of microtubule functioning. But conversely, also learning and memory processes are known to be associated with microtubule cytoskeleton reorganization and improvements. So we can actually improve our own neuronal infrastructure by learning and studying and learning new languages and new skills, which is actually a very important message, maybe the most important message of this series of 4 lectures, that by studying, learning, using our brain, we make it better and make our own cognitive abilities better in the process. All right, so this is another view of the same structure, and next to it you have a micrograph from an actual slice of a neuron across a neuron with microtubules, not so perfectly straight and aligned, but nonetheless generating this dense network. They are roughly separated by about 90 to 100 nanometers of space between them, but densely interconnected by these microtubule-associated proteins, and damage to these so-called maps quite often results in major cognitive impairment. What else do I have to say? Remember, microtubules are 25 nanometers thick. So these, when you look at the image on the right, these structures are 25 nanometers across, and they are separated by something like 90 to 100 nanometers distances. All right. Now, the grand challenge in all of this is to try and understand the hierarchy, with starting with the whole brain, the dimensions of the organ, and which are microscopic, and the 1.5 kilogram in weight, and the dense networks of neurons that are performing various functions, different areas responsible for different aspects of our functioning as an intelligent species. When you go down to a single neuron, then you see synaptic connections, you see the microtubules and the axons and dendrites. One important aspect of dendritic microtubules is that they are antiparallel. Microtubules do have an orientation. There is a plus end and the minus end, and in dendrite they are oriented antiparallel. So one is from minus to plus, the other plus to minus. And you have to also remember that there is an incredible amount of exoplasmic traffic, motor transport taking place, also directionally. So kinesins, motor proteins moving on microtubules, they always do so from the minus to the plus direction, and dyneins in the opposite direction. So you have kind of a highway with divided highway with directions to the plus end and then to the minus end. avoiding collisions, organized traffic actually. And in dendrites, you have these bidirectional lanes of microtubules, which is quite intriguing. And then, of course, you look deeper, and what you see is the building blocks of microtubules. In the panel on the right are tubulin dimers, alpha beta dimers, which are heterodimer, so analogous but not identical proteins, tubulin, and they have great capabilities of forming various structures, not only microtubules but other geometrical structures, sheets and macrotubes and oligomers and so on. But the most important part about them is that they have absolutely unusual electrostatic and electrodynamic properties. So without going too deep yet into the sort of quantum underground that is lurking around, microtubules and their building blocks are very special types of protein structures. One of the most unique properties of microtubules is the so-called dynamic instability. They grow and shrink stochastically, and that's kind of a never-ending process for as long as you supply the energy in the form of GTP, and no other filament in the human body does this kind of organized activity. They actually synchronize that. So that's a deeper look at the organization of the cytoskeleton inside the dendrite here. And in addition to these microtubule associated proteins, the thicker bars connecting the bluish green microtubule structures, there's also the C-termini, which are those flexible peptide chains decorating the surface of microtubules. And they are highly electrostatically charged, and also they attract and interact with both MAPs and motor proteins. So they are very important in the dynamical functioning of these structures. So microtubules do not necessarily directly go to the synapse, but they are indirectly connected to the synapse via actin filaments, which are closer to the cell membrane. But they do actually interact with ion channels, and ion channels, for example, are involved in signaling between cells through calcium waves, and microtubules are very sensitive to the ionic concentrations. They are actually, they can be destabilized by calcium, and conversely by zinc ions can be stabilized, or in fact they require zinc for stability. Here you see kind of a cartoon representation of a microtubule interacting with the calcium ion channel, and there is a motor protein moving on it, and there is some other protein attached to it. And you can see also there's some fragments of microtubules at the bottom are disassembling from it, kind of indicating its inherent dynamical nature as a protein polymer, and that is structural. So we are doing a deep dive into it now, because since late 1980s, I think we've known the crystal structure of tubulin. Now there's a lot of experiments showing tubulin in interaction with different ligands and other proteins, and we know quite well what it's composed of. And here on the left, you see the tertiary structure of alpha beta tubulin heterodimers, and then that is the building block that enters into these thin green lines, which are called protofilament, and 13 such protofilaments wrapped around a cylinder form a microtubule with a cross section, as you can see, which is circular with an empty or void interior called the lumen, and that's one of the most sort of studied aspects of microtubules. What's happening in the lumen seems to be protected. It could be transferring some information from one end to the other. And remember that the length of microtubules can actually span the entire cellular dimensions. They can be tens of micrometers long. Okay, so now there's been a lot of speculations, and this kind of leads us gradually into this quantum biology aspect of it, that microtubules can store and perhaps even process information and signals. I've been involved in some studies showing signal propagation through microtubules, electrical signal propagation, and I mentioned the C-termini. So every dimer has two of these C-termini, which are flexible, charged, and they can be in at least two conformations. One conformation is perpendicular to this, roughly perpendicular to the surface of the microtubule, and the other is bent. So there could be several states. When it's perpendicular, it can actually interact with almost like grabbing hold of something, interact with other proteins, for example, motor proteins or MAPs. When it's bent to the surface, it's not interacting. So that could be several states there. There could be also electron hopping from one positively charged area of a microtubule to another, so several electronic states. There's 8 tryptophans. I think I mentioned that last time when we talked about photosynthesis and somewhat similar effects in tubulin and microtubules. Each tubulin dimer has 8 tryptophan molecules, each of which can be photoexcited. So these could be also, these definitely could be quantum states utilized for let's say quantum computing, if you want to call it this way. And then the entire dimer can actually adopt 2 different conformational states. One is straight, when both alpha and beta bind GTP molecules, and the other is bent when beta tubulin has a GDP bound to it as a result of hydrolysis of GTP and a loss of one of the phosphate groups. And finally, there are very strong dipole moments associated with tubulin, and they can interact with other tubulins, and this could be an electromagnetic way of processing signals. As some of you may know, and this is something known in physics for more than a century, well, actually a century and a half roughly, dipolar oscillations generate electromagnetic waves. So because tubulin has such a large dipole and so many of them, one could imagine that oscillations of microtubules can actually generate electromagnetic waves throughout the cell, and vice versa, can be excited by electromagnetic waves, and that could be transformed into mechanical oscillations. There are some papers on this topic from computer simulations, but nothing yet on the experimental front. And the last one, which we'll talk about in some more detail in a minute, is phosphorylation. Phosphorylation is a very well-known method of biological signaling, and most of the phosphorylation involves serine residues on proteins, and there are quite a few serine residues on these C-termini. So we'll come back to it in a second in terms of some more details, but if just at face value, if you were to calculate how many bits of information a microtubule can store, and there can be thousands of microtubules in a neuron, for example, we'll come to it. There is an enormous amount of computational or memory storage power in microtubules alone. For example, C-termini states could be at least 4 per dimer, electron hopping 4 per dimer, conformational changes 2 per dimer, phosphorylation states 4. This is all conservative, and that when you start multiplying, because each of these is an independent state, you end up with about 128 different states per one tubulin dimer. And in a neuron, you could have about 1,000 tubulin dimers. So that no one had, maybe even more. But anyway, when you do the math carefully, and I'm kind of rushed not to be too careful, but I've done this calculation before, you end up with about 100 kilobits per microtubule or 1 gigabyte per neuron. So just utilizing the storage, memory storage capabilities existing in microtubules. So with 100 billion neurons, now we come back to these estimates of what can and cannot happen in the brain. You end up with the potential for having 10 to the 20th bits of information per brain if all the microtubules were utilized for this purpose, which is probably not the case, but even a small fraction of it would give you an astronomically large number. And now the question is how fast these states can be transformed or can be switched using popular language. State switching would be equivalent to information processing. If microsecond transitions can occur, which is, let's say, quite reasonable for conformational states, microseconds or to milliseconds, but staying with microseconds, you get 10 to the 26 operations per second, which, remember, comparing to the blue gene, it's about 11 orders of magnitude greater, or 100 billion times more than the blue gene, if all the microtubule processing power was somehow harnessed by the human brain. And you may remember that Albert Einstein, I think, quipped that we only utilize a small percentage of our brain's capabilities. Maybe he used more than most of us, but these are the numbers, and there are some specific frequencies. So now we're coming to the question of this time scale. What is the clocking mechanism? Because that's very important. We can see here an enormous potential for encoding information, but how fast can you switch information? That's the processing power. And you know how our computers improved over the last several decades, and all of this had to do with the faster and faster transfer of information and bit switching over shorter and shorter periods of time. So this is kind of a table of known frequency ranges involving microtubules, single microtubules, depending on what sort of physical input is produced in the mechanism. You have different frequencies. So acoustic could be 500 kilohertz, quite slow compared to other ones like ionics. Inputs could produce megahertz frequencies, and conformational could actually be very much faster, up to and perhaps even greater than terahertz. A terahertz is 10 to the 12th. So that would be actually on a picosecond time scale, and so on. You could also consider electromagnetic interactions in the visible range. However, as you increase the scale, typically in physics, the bigger and bigger the object of study becomes, the longer the time scale, or the lower the frequencies. So it's as you increase the size of the networks of microtubules, for example, or then you build networks of neurons, these become slower and slower in terms of their clocking frequency. And we know that the human brain kind of registers, at least consciously registers inputs on the order of hundreds of milliseconds, maybe 100 milliseconds, maybe a bit less, depending on the situation. But that's more or less the time scale. So these time scales here are much, much faster because it's a fragment, smaller. And this is a kind of a dense slide, but it gives you some context in terms of how you can put an upper limit on the processing speed of all these elements. And this is all limited by the amount of power the energy per second that is delivered to the neuron, for example. And as I mentioned earlier, the brain uses about 25 watts, but perhaps only about 3 watts, which is joules per second, are used for information processing. And 1 bit of biological energy, we can assume, costs about 1 ATP molecule. So that's the biological energy quantum. And here's this word, the quantum. So energy here is quantized very definitely. Energy inputs for every process are required. So especially when you want to erase some information and replace it with something else. So if you do this calculation, you end up with this amount of power delivered to the neuron. So divide 3 watts per 100 billion neurons, you end up with the following aspect: somewhere between 10 to the 9th and 10 to the 10th bits per neuron per second is kind of the upper limit, as I can estimate. So a gigabit per neuron per second, that would be pretty impressive anyway, but that's, in my opinion, the limit that we cannot overcome because of the limited power supply. And now, when you bring it down to the processing units, what's a processing unit? If it's a microtubule operating at a nanosecond scale, then you end up with 1 bit per unit of clocking time, nanoseconds. And there may be some other computational elements in the neuron. For example, ion channels could perform computing, or the entire action potentials, and all of these operate at different time scales. So action potentials perform at millisecond time scales, and so the upshot of all of this is that the number of bits per time step per neuron can vary between 1 to 10 bits per nanosecond, or 1,000 to 10,000 per microsecond, 1 million to 10 million per millisecond, or billions of bits per second, depending on the clocking speed. Okay, so I don't know if this is clear, but you can spend a few minutes doing the calculation for this slide, and I think it's correct. So these are the kind of predictions of the processing power of the neuron, and then of course you can multiply it by 10 to the 11th to get the corresponding values for the brain. So this is kind of a hierarchical model of information processing, where you have very few fast modes of information processing, such as those occurring through tubulins. There's 10 to the 18th estimated 10 to the 18th tubulin dimers in the brain. So this is yeah, and you have lots of fast units, but there is a limit on how many bits can be processed and on that time scale because of the power, as I mentioned too. And then you have many slow transitions with few processing units. I mean, few is relative, 10 to the 11th neuron. So 10 to the 18th tubulins per brain, 10 to the 11th neurons per brain. You end up with yeah, these estimates. So by the way, this 10 to the 26th operations per brain is an overestimate, because basically that would fry the brain, and the temperature would go so high, you couldn't cool it down. Okay, so this slide illustrates the various types of energy transfer processes. Some of them we know are ionic waves propagating through microtubules. Some are protonic, we know that as well. nobody has so far, to my knowledge, determined if there is any electronic conduction. We've done some experiments in our lab, not published, but we measured electronic conductivity in dry tubulin. It's much lower than for microtubules, but it's non-zero. So that can also occur. And this is an example of one of these measurements that were published. You can see in the upper right corner a microtubule between two micropipettes with electrodes, and the signal is sent from one electrode to the next over a distance of about 20 to 50 microns, which is the length of the microtubule. This amplitude of the signal is about 200 millivolts, and you can see in these curves on the left in the bottom corner, two curves where you have the injected current in blue, or the input voltage, and you have the collected voltage in red. And you'd right away notice that the collected voltage is greater than the injected. So that's one of these weird properties of microtubules, that actually they act as transistors. They amplify signals. And that could be of technological importance, but also very important to understand that they can actually amplify signals inside the neurons of our brain. All right, so they are very sensitive. Now the question is the brain a quantum computer or just a computer, or maybe not even a computer, but something else? And if so, are we talking about the collapse of the wave function, which could be an effect of a conscious activity? And some people associate the collapse of the quantum wave function with something like a realized thought. You know, we may have several thoughts in our mind, not crystallized, but once you open your mouth, like myself now, all these thoughts are becoming real and vocalized or realized. So from quantum state of mind, which, you know, every few hundred milliseconds, I think I have new thoughts in my mind, and so are you. But then these thoughts become most of the time crystallized. Sometimes not quite. So that will be kind of an interesting, perhaps metaphor, or perhaps a real representation of what's going on in our brains. So as far as we know, these were quantum states. They probably, in physics, we would say right now they can persist for some nanoseconds, but going up to hundreds of milliseconds would be a very challenging task to prepare such states. We don't know how to do that yet. Now, let me switch gears a little bit, although we'll come back to this at the end of this presentation, come back to the question of quantum or not quantum, because our cognition sometimes is unfortunately impaired by various diseases, neurodegenerative diseases such as Alzheimer's, Parkinson's, Creutzfeldt-Jakob's, ALS, Huntington's, and traumatic brain injury, which is a debilitating disease, especially afflicting athletes, you know, in contact sports like football and hockey and boxing. And in all of these diseases, including TBI, there is always pathological. You're a pathologist, so maybe you can correct me if I'm wrong, but one of the known molecular pathologies is actually disruption or damage to the cytoskeleton of neurons, especially pronounced in TBI, but in other ones as well. And if it's not, let's say, broken microtubules or fewer microtubules in neurons, then there is a deterioration of the motor transport on microtubules. So either direct or indirect effects on the cytoskeleton. So whether it's just structure or beyond, we don't know yet, but I think it would be rather unusual if structural damage didn't have any functional implications. And in terms of Alzheimer's, the Alzheimer's disease, for example, there are two simultaneous processes taking place, one of which is the beta amyloid accumulation in the extracellular matrix outside neurons, and the other is directly affecting microtubules, namely hyperphosphorylation of the MAP tau, which forms neurofibrillary tangles inside neurons, and also that leads to microtubule destabilization inside neurons. And you can see here kind of a cartoon representation of what happens to Alzheimer microtubules, Alzheimer brain microtubules. The MAP tau is that red structure that connects between microtubules, and then when it's hyperphosphorylated, phosphorylation is indicated by a yellow circle. MAP tau tends to aggregate into these neurofibrillary tangles, and a consequence of it is this destabilization of microtubules. As you can see in the middle, the microtubule is gradually disintegrating. So in other neurodegenerative diseases, you have typically mutations or some post-translational modifications of proteins, which lead to misfolding of some proteins, such as alpha-synuclein in Parkinson's disease, and Parkin as well in the same disease, and then aggregation, forming pathological clusters of these proteins instead of properly distributed in normal cells. Huntington is the culprit of Huntington's chorea, and so that's also due to mutations. And one of the consequences is disruption of its binding to microtubules, resulting in impaired axonal transport. And there is a list of other diseases with consequences for microtubule or microtubule associated proteins and their distribution and interactions. Epilepsy is also linked to such impairments, ALS, and MS, and Charcot-Marie Tooth disease. There are stalled microtubules in the latter, and mutated dynamin protein, which is involved in the regulation of microtubule dynamics. So, so there are these sort of correlates of microtubular structure and function changes in both pathological and normal states of the brain. But the question is, what is memory actually? You know, we talked about information storage, various states of information. Are they actually encoding for some memories? We don't quite know that, but according to neuroscience, I think memories are formed by changing synaptic strengths, the so-called synaptic plasticity, and the current paradigm involves long-term potentiation. So stimulating the same neurons by the same kind of activity, for example, learning a new language or learning another skill, causes long-term potentiation and facilitates the excitation of such neurons through these synaptic connections, which can be enforced, or if not used, could actually deteriorate. But we still don't know if this is the end of the story or there's something deeper than that. And I personally believe that there's more to it than that. And this is a kind of a summary table of the various predictions about how much memory we can actually store in our brains. I talked about processing, how much processing we can do. John von Neumann at the top of this list in 1950, he was actually one of the pioneers of modern computing, a quantum physicist, but also an information scientist. And he actually predicted that the human brain can store 3 times 10 to the 20th bits of information, which I tend to agree with. I mean, based on some of the numbers I gave you and some of the numbers I'll still give you in a few minutes. Some anatomists in the 70s estimated this to be about the range of the synapses, 10 to the 13th or the 15th. So not as impressive as von Neumann's prediction, but Thomas Landauer in a paper in 1986 actually gave a very pessimistic estimate of only 10 to the 9th bits of information in the human brain, which is, these days you can buy a terabyte of memory on a memory stick. So I think comparing to modern technology, we probably can do better than that. His estimate was based on the assumption that we retain 2 bits of visual, verbal, and tactile information per second. And because the human lifetime is something like 2.5 billion seconds, then he ended up with about 5 billion bits per lifetime. Of course, we forget some things and remember new ones and so on. So there's a bit of a replacement of memories with new ones, but nonetheless, these are the numbers. And so 10 to the 20th, maybe 10 to the 19th. How does it compare to some things you can relate to? Well, apparently the number of words ever written is equal to about 10 to the 16 bytes. Bits and bytes I use interchangeably, but that's not the same thing. 1 byte is 8 bits. So these two numbers actually agree quite well. So number of words ever spoken, 10 to the 19th bytes. And data on all digital media, that was some years ago. So probably is already 1 or 2 orders of magnitude greater than this 3 times 10 to the 19th bytes. Okay, so in terms of the actual physical speculations about where is memory stored in the brain, different theories were proposed. Karl Pribram, a famous neuroscientist, proposed holographic memory, that we actually create 3-dimensional holograms in the brain, and these holograms are recalled when you think about some memories. So actual, almost like a projection, like a movie you can watch in your own mind's eye. Rupert Sheldrake actually claims that memories are not stored in the brain at all, but they are present in the morphic fields, which can extend outside of the human body. Well, it's an interesting hypothesis, but I don't think there's any basis, empirical basis for this. Different kinds of memories exist in terms of animals. That bacteria can learn, and mice can do mazes, and this is actually what and some of the skills are passed down to the next generation or generations, which implicates epigenetics, that not just the genetic code, but also some modifications, post-translational modifications, which are maintained, could serve as memory. Mike Levin from Tufts University in Boston performed some brutal but incredibly interesting experiments where he decapitated worms. These worms regrow the rest of their bodies when they are decapitated, and in fact, after the regeneration of their heads, they were shown to demonstrate persistence of their long-term memories in some kind of trained experiment. So that was published several years ago by Levin and his collaborators, showing that maybe, at least in animals, memory is not necessarily stored in the brain, but could be elsewhere in the body. Now, in terms of coming back to microtubules again, I mentioned that they are implicated in all kinds of remodeling in the brain following learning, and the opposite happens in terms of damage, loss of memories, Alzheimer's. So where can this be? And there were some experiments done on mice, which were trained to perform some tasks, and then they were given or administered an inhibitor of a particular enzyme called calcium calmodulin kinase 2, which is an enzyme involved in synaptic interactions, shown here. This hexagonal enzyme, which is activated by calcium, and then it phosphorylates actual microtubules. So these mice, when were given the inhibitor of that particular enzyme, they lost memories. So there is a connection to this pathway, but how does it link with microtubules? So as I mentioned to you, at the cell level, we believe that long-term potentiation is at least strengthening memories, if not forming memories. But at the molecular level inside the neuron, this may be the mechanism. Phosphorylation of microtubules in axons and dendrites by calmodulin kinase 2 enzyme. And so we did some computational studies following this paper, experimental paper published a few years prior to that. And what we found is that this would actually enable the storage of huge amounts of information on microtubules in the form of phosphorylation. Here's a particular cluster of tubulin. Remember, we're talking about a microtubule with 13 protofilaments and thousands of layers of that. So we're talking about tens of thousands of microtubules and thousands of such clusters. Each cluster here has seven dimers, and each dimer can be phosphorylated in two places, or not phosphorylated. So you have three states. So we call the bits trits. Bits is 0, 1. Here is 0, 1, 2. So three states. And actually, if you each calmodulin kinase has six let's call them legs, because it kind of walks. It's a motor protein also, which works on microtubules, you'll see in a second, and can phosphorylate 6 different monomers of tubulin. So when you do the calculation, each event can actually encode anyway from 64 to 5,218 bits of information, and you multiply it by the number of these hexagonal-like clusters. And it's an encoding which actually is quite robust, because it involves phosphorylation. So it's not something that can be temperature raised, thermally removed. You have to actually dephosphorylate it enzymatically. So it's energy expensive. But these are energy quanta, actually. So in a way, it's a quantum process of energy information encoding. So this is a model constructed by my former student, Travis Craddock, who is now a professor of neuroscience in Florida. And this is a beautiful snowflake-like structure, this calmodulin kinase 2 enzyme. The cross-section looks, beautifully hexagonal. It is actually composed of 3 layers. You have the body, and then you have 2 layers, one above and one below. And it walks with these leg-like motor domains on microtubules. And when you superimpose it on the structure of tubulin in microtubules, it actually makes a perfect match. And I mentioned to you that phosphorylation can involve serine or threonine, preferentially serine, which is in cyan, and you can find these serines on C-termin and also some on the tubulin body. And here's how geometric matching works. In this case, you can see how this snowflake superimposes on the patch of tubulin quite perfectly. And and also matches electrostatically. So it's attracted by electrostatic forces with the opposite charges, negative charge on largely on tubulin and positive on these motor domains of calmodulin kinase 2. So these are the encoding types of structures I mentioned earlier. Maybe that could convey some biological information. We know phosphorylation is a biological signaling method. And again, if you do the calculation in terms of how many states you can change and therefore encode information, you end up with something like 1 gigabyte per neuron. So we are, from a different perspective, coming up with these staggering numbers of information states in individual neurons. And this could also be interpreted as some sort of functional information, because microtubules, as I told you, are involved as traffic highways for motor proteins, moving cargo from place to place in the cell. And these phosphorylation states may actually give instructions as to where to go and where to stop, where to go, where to turn, and so on. So we've actually proposed the construction of universal logic gates and an exclusive OR gates, such as in electronics, using the phosphorylation states and maps. In this case, maps are these yellow and red lines which connect between microtubules. So you are now creating a network, a logical network, building this biological computer, if you will. And back to, this was actually the work I've done with my student Travis Craddock and Stuart Hameroff, and I mentioned Stuart Hameroff in connection with his prophetic book, Ultimate Computing from 1987, and only about 25 years later, this is the paper that we published in 2012. So 25 years after the Ultimate Computing book, we came up with something actually actionable, some sort of implementation of this idea into microtubules. And as you may or may not know, but Stuart Hameroff and Roger Penrose have collaborated for over 2 decades now. Roger Penrose got a Nobel Prize in Physics in 2020 for his work in cosmology, black holes, etc. But starting in the 1990s, he also turned his attention to the topic of consciousness. He wrote 2 best-selling books, very hard to read, but incredibly informative. Emperor's New Mind and The Shadows of the Mind, which kind of stimulated this interaction with Stuart Hameroff, who is an anesthesiologist, and they came up with this theory that microtubules in our brains are actually quantum information processors, or and they call it the substrate for quantum consciousness. Roger Penrose maintained in these books and argued very convincingly that the way the brain works cannot be algorithmic. There are some examples in this book, and I will not go into details. You should pick up the book and read it. But they're very good examples from the game of chess, from some mathematics, from some tiles, and so on, that human brain can solve almost instantaneously some problems that the computer would get stuck on. Couldn't solve them if you designed sort of algorithms, classical algorithms to do it, and that's the basis, and therefore that leads to potential quantum processes involved. Maybe not all of our cognitive abilities involve quantum, but some might, and so that led to microtubules, and that led to the functioning unit tubulin that has these 2 or hundreds of states, some of which may be classical, some can be quantum, and and then and then the question arises, okay, how do you test it? How to test such things, because you cannot probe the brain directly of the human being, or even if you could, this could be very subtle and you would destroy the state. But we know already something which does the job of turning on and off consciousness, and that's anesthesia, which was found, which was what's the word? Is it discovered? I guess invented. It's both a discovery and invention, because we use natural or chemical substances, agents, which produce anesthesia, but also some atoms like xenon and another one. I'm forgetting now the name of the other atom, but single atoms can cause anesthesia. So how does it work? It turns off consciousness, and amazingly, anesthetics. We don't know how it works really. There is no known mechanism. There is hypotheses, of course, that anesthetics interact with membrane receptors in the brain and block them. They also interact with the membrane itself, because they are hydrophobic molecules, and therefore they don't like to be solubilized. They really like to be in the lipophilic environment, and phospholipids in the membrane do the exact job. And but it turned out that of all the detectable proteins in the neurons, 500 or so, tubulin and actin are the only ones which are known to interact with all anesthetics. So that's, of course, indirect evidence. We did some simulations on this. This is a table of different anesthetic molecules, both inhaled and intravenous. Most of them are inhaled. So halothane, isoflurane, enflurane, methoxyflurane, chloroform, fluoxetine, many of them you see here. Yes, argon and xenon and nitrogen. Argon and xenon are single atoms. Nitrogen is as a biatomic molecule, but there are also propofol, which is IV injected, and ketamine. So there is no commonality in these structures chemically. Virtually no visible similarity, as I mentioned, from a single atom to two atoms to some aromatic molecules and some very different chemically structures. But they all interact with tubulin, and tubulin is therefore a potential molecule where the action of anesthetics takes place. So we did some simulations, computer simulations involving tubulin and anesthetics, and we found binding sites for anesthetics. Again, this was the work of Travis Craddock. He did a fantastic job for his PhD. He found 47 distinct binding sites. So anesthetics bind weakly, but to a lot of places on tubulin. And in particular, some sites are well known, where colchicine, vincalcaloids, and GTP binds, and some other ones. They all have actually a largish dipole moment, and some of them bind close to tryptophans. I mentioned to you tryptophans a few times already. There's something about tryptophans because they can be photoexcited. Their electronic states can be excited, and then they interact through dipole-dipole forces. And so anesthetics can, those at least that bind near tryptophans, as shown here, the tryptophans are in blue on the tubulin dimer, 2, 4, 6, 8, 8 of them I mentioned a few times. And then the anesthetic molecules, I think this is isoflurane here. It's they bind to several of them. There's 1, 2, 3, 4, 5, 6, 7, 8, 9 locations where they can interfere with the tryptophan network of interactions. So if tryptophans are involved in some kind of a quantum state, because we know we can excite them by photons, so generate quantum states of tryptophans, they can be possibly interfered with by the location of anesthetics nearby. And so that's kind of an ongoing effort. We currently perform experiments on exactly this topic at Princeton with Professor Greg Scholes in chemistry, who is one of the original discoverers of quantum origins of photosynthesis, and now we are working on trying to demonstrate tryptophan interactions through quantum excitations, and how that can be disturbed by the presence of anesthetic molecules. So that project is funded by the Templeton Foundation, which supports research in consciousness and especially quantum effects of consciousness. So we'll see. The project has about 6 more months left. I cannot disclose too many results before the end of it, but there are some encouraging and some are still to be determined. So this is the dipole-dipole interaction energy between tryptophans and their locations, and in one of the more recent papers with Travis Craddock and Stuart Hameroff, we predicted that the presence of anesthetics will affect the interaction energy between these tryptophans, depending on the strength of the anesthetic molecule, the potency, and it's with a very high correlation coefficient. This is a plot for 1, 2, 3, 4, 5, 6, 7, 8, 9 anesthetic molecules in these red dots, and then there are some similar structures of molecules which are not anesthetic. They do not fit into that line, which is a good result, because we can distinguish between anesthetics and non-anesthetic molecules by how they interact with tryptophan dipoles. And this is a shift in terahertz of the frequency, correlating with the oil gas partition coefficient. If you know, at the end of 19th century, beginning of 20th century, there was a finding published by Mayer and Overton which indicated the basis of anesthetic mode of action, at least at the cellular level, namely that their potency is quantified as so-called MAC, or minimum alveolar concentration, is correlated with the oil gas partition coefficient. So the more oil-loving the molecule is, the more potent it is as an anesthetic. And you can see here on the right this famous Mayer-Overton plot, which is almost a perfect line, how these anesthetics scale with their potency vis-a-vis lipophilicity. We see maybe not as perfect a line, but this is more at the molecular level in terms of interactions with the tryptophan oscillations. Now, I will slowly wind down this talk, but there are some other molecules which affect our state of mind. So I talked anesthetics really turn off consciousness, but some other molecules affect our state of mind, conscious states. Some of them may produce excitations or altered state of consciousness. Some will give us some feeling of pleasure, caffeine, chocolate. You can see the almost identical molecules, but some others may be dangerous, and you can see LSD here and ecstasy and heroin. So the idea is that we haven't done this, but what I have done with my postdocs in the past, but we never published these results, that pretty much all of these so-called psychoactive compounds bind to tubulin, and they bind even more strongly than anesthetics. Whether it has any meaning in terms of their mode of action, molecular mode of action, we don't know, but they definitely do bind in several locations, and the locations differ on tubulin depending on which one you look at. So they come into different categories: stimulants, hallucinogens, depressants, and antipsychotics or tranquilizers. And it would be interesting to see whether they might actually. We haven't finished the study. I only know that pretty much all of them bind to microtubules and tubulin, but we haven't done any sort of mechanistic estimate of the effects on the dynamics of these quantum excitations in microtubules. So this is still early stage, but there are some indications that that there is a lot of activity inside the neurons. You know, some of the more established projects on the basis of neuroscience, such as the Human Brain Project in Switzerland, supported by the European Union with 2 billion euros of funding, stop at that point. I mean, they do assume that neurons are basically the elementary units of cognitive processing in the brain, and they represent them as passive elements, basically switches. But we look deeper into it, and in this talk today, I try to convey a message that there may be actually a whole new world going on inside neurons with all these complex structures that interact with a lot of molecules, and we know that they are important in various diseases, both for neurodegenerative aspects, but also, which I didn't talk about, in diseases such as cancer, where cells divide. Now, let's see if I can. Okay, I will end with something completely hypothetical, and that's not I would say hard quantum biology, but something where I think metaphors from quantum physics can be useful, and I call it toward quantum psychology. So we look at the mental state, not at the microscopic elements of neurons, as I kind of tried to convey the message in this talk. But now we switch gears and go to the state of the mind, the entire state of the brain. So in quantum mechanics, there are a lot of attractive ideas or concepts which psychology and psychiatry could actually benefit from. And one of such ideas is the concept of second quantization. Basically, you have operators which take you, I'm almost done at 5 minutes and we'll be done, which take you to higher energy states, or vice versa. They can take you to lower energy states. So these are called creation and annihilation operators. And I'm kind of proposing that they can be useful for the classification, characterization of mental states. More and more excited or depressive states. You can create a ladder of these energy states, mental energy states. Likewise, we can introduce the concepts of quantum statistics. And we talked about this in the first lecture, fermions and bosons. Fermions are exclusive quantum particles so they can occupy only one state, these particles individually or none at all. And for bosons, you can have many states. So why not use it for normal states of mind, for fermions, or multiple personality disorders, where you can actually superimpose many states and create a superposition like this. And then there is, of course, commutator. One of the canonical aspects of quantum mechanics is the lack of commutativity. So commutators, two operations give different results depending on the order in which you perform them. Two measurements. So the measurement of position, velocity, will give you a different result in quantum mechanics depending on whether you measure position first and velocity second, or vice versa. The same may be true of all psychological tests or queries in psychological examinations. They may not commute these things. So the order in which you ask questions the patient may affect the answers. And then there is the more exotic aspects. I haven't even talked about it, but commutators of these operators, some new types of algebras, so-called Q-deformed algebras, where you actually have the results of this commutation distorted depending on the parameter, which is called the Q parameter. And so maybe in some mental states, pathological mental states, you could introduce these kinds of distortions from normal kind of operations. And finally, the concept of coherent states. So states which are minimally diffused, if you will, such as laser states. You could probably relate it. So this is all metaphorical. Don't take it literally. But I'm proposing these concepts to be useful in psychiatry for maybe introducing more quantitative analyses. So coherent states could relate to things like meditation or lucid dreams or even mystical experiences. Okay, so let's see. I think my time is up. I've actually talked to some psychiatrists about all these things. So there is some, and I've given a talk at the annual conference of American Psychiatric Association, I think, about this, and there was some resonance in some of the people that quantum concepts could be beneficial to the improvement of some of the classification systems in psychiatry. So anyway, I'm throwing this out as a potential advertisement for collaboration with people in these fields, where such as psychology or psychiatry, where I think there are some barriers to progress. Maybe some of these barriers are because of the fact that the field has not been open enough to other influences. So thank you very much for your attention, and I will be very happy to answer any questions you may have. Thank you.
Yeah, thank you very much. So any questions?
How do I?
There we go. Okay, let me try putting us in an immersive state here. All right, there we are. Yeah.
Okay.
Okay, any questions? So,
Hello.
hello.
So I have heard that oftentimes you can't tell the difference between a normal person and a psychologically ill person from looking at a brain scan alone, or there's no difference to be detected between them. But do you think that quantum mechanics could potentially solve this problem and explain why people are depressed even when their brain looks the same as a regular person's brain?
Yes, I think that's a very good question, very appropriate for it, because first of all, these brain scans, whether it be EEGs or MRIs, I think their resolution is not that great. I mean, their resolution at best is like a millimeter on the spatial scale. So I don't think we get to the bottom of this. I would look for more dynamical analyses rather than scans of structural. There may be no structural changes. There may be simply more operational changes. And in fact, I think I'm not really probably competent enough to comment on this, but all of us may experience some abnormal states at different times of our lives. So it's not so much about structural damage or structural changes as perhaps the dynamics, and some of us may have potential for transitioning into these dynamical states, which are let's say obsessive compulsive behavior or attention deficit disorder, things which are basically such as quantum states in which you can get trapped, but you need something external stimulus or some maybe perhaps pharmacological agent that will take you out of it. So if these states can be changed as a result of let's say a pharmacological intervention, for example, or maybe some other behavioral, then obviously you wouldn't see through imaging any. I don't know if it's obvious, but possibly I would say you wouldn't be able to see it through imaging. But maybe there are some, through some more subtle, like correlation analysis. Quite often, dynamics, dynamical analyses will provide you with a better insight into these states than sort of static snapshots, if I understand correctly your question. But
Yes.
Definitely, it's a great area of investigation, in my opinion.
Thank you.
Okay.
Yeah.
Hello.
Yeah.
I have a follow-up to that question. So at the end in your proposed diagnoses of mental illnesses based on quantum states, how would, in a living person, how would you even examine the quantum state of their brain structure?
Well, I think we need probably, first of all, to collect a lot of empirical data, but what I proposed, I mean, this is more or less to kind of mimic the path that physics took to uncover the quantum states, and in particular the lack of commutation properties. So perhaps I think in some of the standard testing for various mental diseases or I don't know if disease is the right term these days, but maybe disorders or pathologies, you could try and classify or try and understand the differences between questions asked in different order, and maybe that's what I kind of hinted at in terms of this deformed algebras, and in some cases maybe the differences would be subtle and some more pronounced, and depending on the type of currently understood type of pathology, you can end up with classifications which are more quantitative. Again, I'm kind of out of my depth a bit. I'm just proposing some concepts and hoping that some of the colleagues in these fields would collaborate on that, because because I'm not an expert in that area, but I do feel that mental states are really very similar in nature to quantum states. They're sort of dynamical fluidity, and the fact that quite often I'll give you one example, and nothing to do with pathology, but even just normal way of thinking. If you just think about something, right, you may be convinced that you're right, and this is the right solution to a problem or something. And then when you verbalize it, you suddenly and talk to somebody. So this interaction, which would be like quantum wave function collapse, suddenly it turns out that the answer is different. This is not what you thought when you try to say it, and somebody is even just listening to it. So these kinds of observations, I think, might be useful in trying to think about what quantum physics can contribute to psychology or related fields.
Thank you.
Okay.
So any other questions? Okay, well, thank you for 4 great lectures, and we look forward to it next term and the term after, and on forever, right? So anyway.
As long as you run it, I'm here.
Sounds good. Okay, thank you, Jack.
Thank you, guys. Thank you.
Bye-bye.
Bye.